introduce yourself on the chat box and, for us to be able to know who's here, a little bit about your background and interest. Welcome, Kayla.
Thank you so much.
We also have Richa. Hello there. Okay, we're going to start here shortly. Okay, we also have Kurt Ragnar from University of Nevada, and we've got Spanden, Jason. Welcome, everybody. Okay, so you hear me okay, guys? All right, so let's go ahead and start. All right, so welcome to this overview session on the summer 2022 Omics Logic virtual training programs. And the session will give you additional details about the program registration, levels of complexity, desired outcomes that you might be able to achieve over the summer. We have a range of options that you can look into further and register by the end of second week of June. And the programs we will be offered virtually around the topics of coding in R and Python, infectious and chronic diseases, as well as a research project program that will help you complete a research project leveraging public domain data, big data infrastructure, and training and support from our mentors. Now, because ultimately the methods that are best learned in the context of real case studies prepared by our network of mentors and collaborators that illustrate how various bioinformatics methods and omics data types are used in current research in medicine, biotechnology, agriculture, and even astrobiology. The projects are prepared by taking publicly available data sets and using them to explore and understand both the research challenge and the analysis path to extract meaningful insights from complex omics data. The Omics Logic portal gives students options for analyzing a given data set, code-dependent paths or code-independent paths. Now, these data sets and associated publications can be used in several ways. You can learn bioinformatics without any coding at all. For that, in each project, we explain the analysis pipelines on the user-friendly T-Bioinfo platform for big data analysis. You can learn the scripts behind many of the steps, learning to wrangle, clean, visualize, and analyze data in popular languages like R and Python in order to create your own analysis. Finally, you can hear about the projects and their research applications in our mentor-guided programs, where experts share their experience and support research projects developed by you. Now let's start by signing up to the Learn Omics Logic portal. If you can please go to learn.omicslogic.com. This educational portal will help you get started on your bioinformatics journey. I'll go ahead and post the link. Bear with me. Simply open any browser, go to learn.omicslogic.com. For those that have not signed up yet, feel free to sign up. And if you have any questions or issues in signing up, please feel free to reach out. Now, for those who have already created their profile, can you please type yes on the chat box so we can see who already has a profile that we have here on the webinar? Okay, perfect. We have Nader, we have Catherine, Ashley, thank you. we'll just give a few minutes for those people that have not signed up yet to quickly create their account. And you will see there on the website, there's the quick sign in using Google, email, your Facebook, Twitter account. So feel free to use those options as well. All right. Now feel free to just put on the chat box if you have any issues, okay? Now, once you have created your profile, we would like to encourage each and everyone to update your profile in order for us to track activity and issue achievement badges and or certifications. Now, the complete profile includes a profile picture, and you can add an avatar or your university logo if you don't wish to add your own photo on there. Connected social media accounts, you can use Facebook, LinkedIn, Instagram, and Twitter account. And a biography. If you can please add a brief bio about yourself, including your research interests. Once you have signed up, this is actually what you will see on your profile page. And there are 4 tabs that I would like to discuss.
We actually don't see your screen. Can you reshare it, please?
Oh, sorry. Okay, do you see my screen now?
Yep.
Okay, apologies for that. Okay, so this is the profile page. Once you have completed your profile, you will see the photo on there, quick bio, and there are 4 tabs in which we will discuss as we go through the slides. The activity tab is where we're currently on right now. This is where you will see for each activity that you are going to complete as soon as you enroll or start a program or a course. And then there's also the courses, the programs. You will see there different tabs respectively on the programs that you are assigned to, and the certificates tab in which wherein you complete a certain course, you will be able to view your certificate and be able to download. So like as I mentioned, a successful completion of a program, you will be awarded with a certificate, and you'll be able to access your certifications on your profile. And this is how it looks like. Okay, so after you register, you will be redirected to the course tab. And here you can search for any course title and use the tags to find series of resources around a topic, or choose one of the collections down below. Now, in this video, we will look at the Getting Started collection. And I also would like to add that these Getting Started courses are free, and for anyone who wants to get into the bioinformatics, and you're welcome to share with your network to take these courses. Under the Programs tab of your profile is where you find the assigned programs of your choice. And each program has a scheduled sequence of events, and every event has recorded sessions and assignments designed around that topic. Each activity you complete will get points, which are for completing the course, running a pipeline, and these points are accumulated. So you'll see here, whenever you complete a course, it's 500 points. Running a pipeline is 100 points. When you complete a profile, it's 100, and group activity is for 10 points. In this program, you will also be taken through project examples on the Omics Logic project portal, which will help you understand the practical applications of various omics data analysis and learn how it will be applied to answer research questions in hand. Finally, designing your own bioinformatics research project, indeed. During this meeting, you will be able to learn about the upcoming training programs offered by the National Association of the IDEA Principal Investigators, or NEPI, on bioinformatics. We will focus on 3 programs: bioinformatics for infectious diseases, genomics and transcriptomic data analysis to study viral and bacterial pathogens, as well as host immune responses, transcriptomic data analysis for chronic diseases such as cancer, metabolic, and many others, bioinformatics using R and Python, getting started with sequence and structured data analysis in R or Python. And in addition, we will be sharing more information about the Omics Logic Research Fellowship, a program designed to help young investigators complete a full bioinformatics project over the summer and present it during our virtual summer research symposium. To achieve this goal, we use a combination of online sessions, self-guided study materials, and practical assignments for an immersive experience that has proven to be effective in our well-known and respected Omics Logic programs. Now, talking in detail about the upcoming summer programs, I would like to invite Elia Brodsky. Elia Brodsky has a background in applied mathematics, bioinformatics, and biomedical data science, and he has extensive experience in big data project management in academia and industry. He is the co-founder and is the CEO of Pine Biotech, a company that develops and commercializes data analysis software solutions for life sciences. Since 2014, the team under his management is providing commercial and research support to the numerous academic projects. I will pass on the stage to you, Elia. Thank you.
Thank you very much. I'm going to share my screen. And briefly, what we will try to do in this portion is describe, first of all, how to get started, and then what these different types of programs will include. But I think that we already did a number of presentations about the general content. And so if you have any specific questions, maybe you have some, specific idea about what you anticipate, and you want to make sure that that's what you're going to be actually doing. Or if you have any other questions, feel free to put them in the chat. I will be looking at the chat and we'll try to answer all of those. Before we jump into this, I want to ask Farhana if you have anything that you wanted to share with those that are from the LBRN or LSU students or faculty. And I will unmute you. There you go.
Hello, everyone. Good afternoon. I hope you all are having a great day. Can I share my screen, Ilya?
Yep. Okay, let me know if you can do that now.
Okay, can you see my screen?
Yes.
Okay. So hi, my name is Farhana Musarat. You all can call me Farhana. I'm the program coordinator for the Omics Logic LSU Biomed Summer programs. I'm basically a molecular virologist and working as a postdoctoral researcher at LSU Baton Rouge at the School of Veterinary Science. And I will be assisting mostly the LBRN and LSU members with registration and any other issues that they might have. So these are the courses that we are offering for the summer. And Ilya already described these courses. And the question is, who is eligible for this course? And the answer is, it's actually everyone. So this course is open to all. And anybody who are in USA or any other countries in the world can take these courses, and the courses are available for a fee. However, we are offering this free registration for a limited number of people. And the criteria is you have to be a member of LBRN or LSU School of Veterinary Medicine or NAPI. LSU campuses members are welcome to take the course also. However, the course will not be free for them. So how do we register for the courses? I have been getting a lot of emails about registration process, and I think I need to explain it very clearly. So the registration process includes 2 steps. One is the pre-registration, and the other is the registration. So the pre-registration process includes opening an account in the Learn Omics Logic portal, which is the educational portal, and all your course materials will be accessible there. And before that, you have already registered for the program in the edu.omicslogic.com/LSU Biomed. So once you register here, once you click the register for the program here, you actually submitted the application to us. And when you open the account and create an account and update your profile, just what Japs and Ilya showed, you already are in our system. However, for LSU and LBRN members only, we need additional steps to complete the registration. This step is not applicable for NAPI. So for LSU and LBRN members only, to complete the registration, you have to make sure that you have an account in the iLab. So once you have created an account there, you will have a login information, username and password. And when you log in here, you will see a drop-down menu here on the left top corner. You click here and then you will see the core facilities. And then you will select the CEDAR core facilities and services on the left. After that, you have to click on the request services on the right side top corner. So after that, you will see training module sections where you will see all the names of the courses that we are offering for summer. So in this training module with the red asterisk mark, you have to write down what are the courses that you are interested in. So you have to write down the names of the courses in this white section. So once you are done, you will see here a fund number section, but you don't have to put any number here since the LSU and LBRN members will be sponsored by us. So what you can write here is just write whether you are from LSU, LBRN, or LSU School of Veterinary Medicine, and then just click the submit request to core. And that is how you will complete your registration. So if you have any other questions regarding the registration process, please don't hesitate to contact me. My email address is, I'm going to write in the chat section. It's fmusar1@lsu.edu. So I will try to answer any other questions that you have, or you can write it in the chat section or shoot me an email later on, and we will be happy to answer and try to help out with the registration process. So that's all, Ilya.
Thank you. Yes, I think there is a question about the link for iLab and some other questions in the chat. So again, I think Farhana is going to stay on, and if you have those questions, she could help you with that.
Yes.
All right, great. So again, that was specific to a number of people. There are a number of, again, free licenses especially if you are from NEPI. So how do you know if you're a part of NEPI? There's a website, nepi.org, where you can see the map, and all of the 23 idea states are listed on there. If you're from one of the academic institutions in one of those idea states, you're most likely a part of NEPI. What you need to check is whether that institution is listed, because NEPI is an organization that brings together PIs funded through the NIH IDEA Institution Development Award. So if you receive this information from your core, you probably have access to those free licenses. Okay, so what I will try to do in the next few minutes is briefly explain how this training is going to be different from what most of you are probably used to when it comes to online coursework like Coursera. What are the specific benefits of taking this training, and how will you be able to use some of the skills or resources that you will gain access to throughout the summer? So first of all, as already mentioned by Jabs in the beginning, we try to assemble training materials, a number of data analysis tools, curated project data sets, so data sets that are ready for analysis, and interactive sessions that some of them are going to be available as recorded sessions, or if you're available during that time, you can join them live. And this is to make bioinformatics more project-based or oriented around case studies. So bioinformatics is a very broad and dynamic field. There's a lot of new things that come up all the time. And so it's very difficult to imagine someone kind of comprehensively understanding bioinformatics in only 3 months. And so that's why instead of offering to kind of train you and give you everything you need to know bioinformatics, we're saying this is oriented around case studies. How is bioinformatics used in cancer studies, specifically using transcriptomic data? Or how is bioinformatics used in the study of infectious diseases? For example, if we want to understand genomic and transcriptomic data to study the pathogen and to understand the host responses, right? So those are the case studies. Again, those of you that kind of work already on some of those similar projects, this is the opportunity to take your project and just analyze that data as you go along. And that means that we're going to be dealing with a diverse group of individuals, that some are faculty, very independent, driven by maybe research objectives and questions that they would like to get answered, as well as some undergraduate students that might be new to all of this and are just trying to get started. And so that's why we're going to be offering a number of these different resources so that you can explore the ones that you need on your own time, but also have access to lectures and mentors that can actually help you and guide you and help you overcome technical issues that you might face. So what we've done over the past few years is we have collected industry and academia lectures from a number of organizations, including different NEPI associated organizations, and kind of prepared a basic package. And that basic package includes genomics, transcriptomics, metagenomics, epigenomics, introduction to data science, and kind of collection of different project examples. In addition to that, we understand that a lot of people might come from a biology background, and they might not be prepared for the technical demands of both learning how to program a specific analysis, as well as setting up that analysis to run on a specific infrastructure, like your own laptop, or maybe your core high performance computer or something like that. And so because of that, this program comes with another important resource called the T-Bioinfo analytical platform. Now, the platform offers a semi-guided experience where you have a number of predefined paths that connect independent scripts into a pipeline. And they come with a lot of computational resources. For example, every account will come with 5 terabytes of storage. You can also at the end request for that particular pipeline to be submitted into a list of containerized pipelines that you could deploy elsewhere. And most importantly, you get a lot of support. So if you want to really understand how it works, what are the parameters, how do you change them, how do you run it for this type of data or that type of data, this is what the programs are going to be about. It is to provide you with that kind of technical support. For example, how does this achieve learning goals that we're expecting to kind of arrive at after this program? In many cases, you are probably familiar with figures that represent analyses like statistical analysis, maybe principal component analysis, or maybe differential gene expression, or you might be familiar with mapping and visualization of map reads on a reference genome. So the platform will combine those kinds of analyses into an experience where every point in that process will be color-coded and will come in with an explanation so that you can understand what you are doing, change parameters if need be, and also remember how did you achieve a specific result, so it makes that analysis reproducible. Now, in the program, we will use both the platform and then we will look at what do some of these individual buttons do? How do they actually take in some data that was already prepared for that stage? What type of data do they produce? What does that data look like? And what does the analysis try to do? How does it work? And so because we have multiple comparisons in each logical step, you will see how by looking at the analogous analyses that are available, what are the differences and how might you choose the right type of analysis for your specific goal. I'm going to pause here because I have a couple of questions I'd like to answer. So question number one from Anelia: if you are not a part of any of the funded programs, how do you register? I will share again links where you can purchase the program independently, or you can go to your PI or to your university and say, I would like to take this training over the summer. And in a lot of cases, they will offer to pay for that or maybe support you partially. And the second question from Ashley was about this recording being available. Yes, this recording will be available after the webinar, and also is going to be on our YouTube channel. So as I mentioned, beyond this logic, it's also important to at least understand how code works and what are some of those steps that you can take to get started with coding in R and Python, two of the most popular languages in scientific computing. So what we will do is we will actually use several of the examples after the big data is processed and we've kind of distilled it to structured data or tables. We will look at what is the context. So how do we ask questions about a particular data? Then what is the logic? How do we actually understand the steps to arrive at a particular result? Then we will try to practice, and then hopefully that will prepare you to be completely independent, both relying on big data analysis tools, which you will find many of those are available today, but also if you would like to take your analysis further and kind of take a look at becoming more independent and being able to write such analyses yourself. So now let me talk briefly about three of the programs. So the first one I'll talk about is biomedical data analysis using R and Python. This is a program designed for those who are interested to learn how to write code. So in many cases, you will have a particular type of data that is already structured. And for that structured data, you either want to perform a statistical analysis, exploratory analysis, maybe you would like to also use machine learning. So that is what this program is going to cover. First of all, for those of you that are not familiar with R and Python at all, there's a way to get started, right? And so what we've done on the portal is we've taken some of the basic principles like dealing with objects, loading data into an environment where you can analyze data. looking at specific data sets and understanding structures like matrices, data frames, etc. So those are the things that are going to be covered in that introductory course, and that is available completely for free. If you haven't started with coding, we highly recommend it. We'll help you kind of extract meaningful insights out of complex data, visualize and present them. But beyond that, we're also going to go into kind of a high-level overview of what data science is and how is it applied specifically to biomedical projects. And here we will have two tracks. One is BioML in R and BioML in Python. You can choose either one. And you will see how these are used today when analyzing complex data, but also you will be exposed to a number of standard or what we call conventional machine learning tools, methods that are going to be very helpful to deal with noisy data and data that contains a lot of variability. For example, patient data that contains a lot of variability. So here is a brief overview of the topics that we will cover. We'll talk about data wrangling, how to load, transform, and explore a data set. We'll talk about machine learning and statistics. What is the difference? How do they kind of compare to each other? And why high throughput data presents some additional challenges for these methods? What is the general kind of distinction between unsupervised and supervised types of analysis? How can we use machine learning for data visualization? Then we'll talk exclusively about clustering and another session about classification. We'll talk a lot about how these methods, not only what types of methods there are, but how are they used in example projects. And we'll try to address some biological questions that you might have. So for example, what is the bigger difference between different tissues, or is it between normal and cancer tissue, right? So we'll kind of use those methods to try to connect it back to the types of questions that you might have. Then we'll talk about feature selection and signature patterns, how we can define a particular signature pattern, for example, a marker, and then how can we understand what that marker actually represents, what feature is it connected to. We'll talk about regression and generalized linear models and deep learning and how deep learning is used. So all of these are going to be covered in a live session. So kind of like what we're doing today. There will be someone speaking, there will be questions will be answered, there will be some exercises, but then you will also have a number of examples and additional projects that you can practice independently. So what is that going to look like? Here's an example of a course. This is a course on PCA. So how do you actually produce a plot, a 3-dimensional or a 2-dimensional scatter plot to understand variance in your data? You go through, first of all, understanding the problem. Why are you using PCA in this case? Then you will learn the syntax of the code. So how can code be written to actually perform this kind of analysis? Then you will be given that code that you can run, and you will be able to modify outputs. So in one case, you might produce a table. In another one, you might produce a visual output, etc. Then you'll be able to play around with that code and know that you can modify settings. And this is very important because in so many cases, once you know that there's a library that you can use, you just need to modify it to apply it to your data and then make it do what you would like it to do. It is a very liberating experience, and you can start going through a lot of examples that people have posted online and read that code, understanding what it was doing, and know how to modify it to apply it to your problem. And then finally, once you go through several examples like that, you will be given some challenges. And those challenges will come as an interactive notebook, and they will essentially be an assignment. So you will start with some beginning. For example, you've learned the syntax, you've applied it to a particular data. Then the coding challenge will say, okay, write this code. Maybe there's a missing portion, or maybe write it from scratch. or maybe even apply it to a number of different examples that are not trivial. They will need some thinking to overcome some issues that you will have. And again, as a result, you will have an interactive notebook. That interactive notebook will be shared with your mentor, and your mentor will be able to suggest some maybe technical issues, how to overcome them, or maybe how to use other types of methods for whatever challenge you are facing. Now, importantly, we will look at some of the ways that these methods actually work and what needs to be done with those methods to overcome common challenges like missing data, maybe unbalanced classes. We'll talk a lot about evaluating the accuracy and precision, how to trust your model, how to deploy your model after you've trained it. All of those kinds of questions is something that we will focus on and kind of see real world examples. So what are the outcomes of such a program? The outcomes of such a program, first of all, some technical skills and kind of reading papers, looking at the methods and knowing what they mean, but also putting them into a logical scheme so that you can kind of go from experimental design and all the way to interpretation through the lens of the analysis process. How the data is prepared for analysis, how is it explored to eliminate maybe artifacts, batches, and issues with the data, how to then perform actual data analysis, choosing between maybe a model that is statistical or maybe a machine learning model, and then how to produce inference, something that you can interpret either in the context of that particular experiment or something more generalizable, something that you could kind of build upon to understand something about the general population. So again, what we will do is we will use some case studies. And all of these case studies are going to rely on omics data. So we will look at diagnostics and cancer biology, single cell and tissue composition, and we'll also look at microbiome and infection. And you'll be able to choose a number of curated data sets that will be provided to you to kind of play around with and develop a project of your own at the end of this course, at the end of this training program. So again, if you have any questions about this particular program, any of the topics that I just mentioned, please add them to the chat box. I'd be happy to answer any of those questions. And I'll just give a moment here if anyone has any questions. Okay, so again, how do you register for this particular program? The way you register is dependent on which organization you're from. So you could be from 3 organizations: LSU, School of Veterinary Medicine. The second organization is LBRN, Louisiana Biomedical Research Network. And the third organization is the National Association of Idea Principal Investigators. So based on which organization you're from, if you were not present in the beginning of the webinar, we kind of went through all the different options, how to register, but you definitely have to create an account on the Learn portal. And once you do, please email us. And you can email to a number of emails here. So Farhana shared her email, or you can simply reach out to this email right here. We'll be able to direct you on your next steps and even have a call to discuss your specific questions. Okay, so Raquel is asking when the course will start and end. So this is a summer program, and so it's going to start June 15th all the way until August 31st and extended until September 15th. So you will have a full 3 months to work through all of these different things. There's going to be a defined schedule of live sessions, but all the live sessions are going to be recorded and all of them will be shared on the program page. And I will go over the program page at the end of this presentation. Okay. So the second program is going to be more focused on transcriptomic data analysis, or RNA-seq. And we're going to talk about bulk RNA-seq, when you extract your RNA from the whole tissue and you kind of analyze levels of gene expression and compare them between different groups or creating different contrasts. But we'll also talk about single cell RNA-seq, when you extract RNA with a barcode that can make it specific to a cell. And so this is an exciting field with a lot of new methods that are becoming available. Not all of them are simple to use, and in many of them, you need to use them knowing how they work, so how the data is prepared, but also what are some of the standard procedures and considerations about the assumptions that we have and about the conclusions that we will have from our analysis. So we will talk about first of all, the biology of transcription as a proxy for translation. How can we actually use the abundance, the quantity of RNA of a specific type to infer how this will turn into a specific protein? So this is kind of the most common use case, right? We want to identify some functional state, and we say that the mRNA is the proxy that allows us to accurately identify which genes are actually, which protein coding genes are being expressed. And that is why we think this leads to some functional change. Now, there are several different methods to measure and quantify RNA, but we will exclusively focus on next generation sequencing. And for next generation sequencing, there are several levels that you can kind of use that data to analyze what's going on. So one of them is just to look at genes, and these are protein coding genes. Another one is to look at alternative splicing events, where you have multiple exons that connect together, and you would like to understand how that leads to different transcripts or different isoforms, so to say, of the same gene. And so that oftentimes is a very useful technique to be able to understand what is actually going on, because not in every case, loss of function is associated with a low level of expression for a particular gene. So we'll talk about the differences between gene and transcript level analysis, but we'll also talk about how to look at that detail by making sure that we are actually developing a transcriptome from our own data and only then going through the process of annotation. Now, we will try to apply this in the context of a biomedical research project. And so we'll talk about how bioinformatics allows us to essentially connect clinical and omics data, how it allows us to address questions around diagnostics and application of therapeutics. So we will kind of extend our understanding of this expression of genes, isoforms, and proteins, translation into proteins, to then ask more biomedical type questions from the data that we will explore. The program will have several different sessions. We'll start from just the basic logic of what I explained. How is that processing done? And what are the important steps in that pipeline? And the important steps are going to be how the data is prepared with minimal artifacts, so that our conclusions about it at the very end are actually reliable, something that we can trust. And so we'll talk about different methods of data pre-processing or cleaning. We'll talk about mapping. How can we map on the transcriptome, on the genome? How do we actually identify those alternative splicing events, etc.? And then we'll talk about how can that data be quantified and used to represent a difference between conditions. So in the most simple experiment, you will have several replicates in one condition, several replicates in another condition, and because they have been kind of produced in a controlled way, you can make biological interpretation based on these differentially expressed genes. And we'll talk about plotting, volcano plots, and things like that, but also about downstream interpretation. How do we connect those differentially expressed genes with gene sets, like pathways, gene ontologies, etc.? And how do we make sense of what's actually going on? Now, to do that, we will have to cover all of the steps from the basic alignment, as you see here, with the reads aligned to the reference genome, to then quantification, analysis of variance, and understanding of whether it is the right approach to be able to compare multiple conditions. And how do we actually go about more complex data sets where instead of just two contrasts, you all of a sudden have many? How can you analyze that kind of data? And that is, again, bringing us back to that question of data mining and classification, really being able to work with larger and more complex data sets and extract meaningful insights by using nonlinear methods of machine learning and feature selection. So then we'll also talk about single cell transcriptomics. So an exciting technique becoming very, very popular. Many cores are now adopting 10x genomics and other machines to be able to produce single cell data. Question is, what can we do with it? And how do we do it so that we are in control of that analysis and we know how to ask biological questions and actually get answers to those? So to do that, we will talk about typical approaches. So the standard output being the UMAP or the t-SNE presents us with a clearer picture that unfortunately is not trivial to explain, because it really contains a lot of information that has been adapted to a particular hyperplane, and it's difficult to connect back to biology. So we will look at how we can use such methods of dimensionality reduction to start asking biological question, going back to the statistical comparisons that we've explored in the bulk RNA-seq course, and how do we then can also ask additional questions about, for example, pseudotime. How can we track trajectories where gene expression levels actually represent a continuous process? Then we will look at the full pipeline again, from understanding of what is in our data to then being able to ask specific questions, and then all the way to downstream interpretation. Now, all of these are going to be given in the context of several different project examples. And we'll talk about patient-derived xenograft models, where we will compare tumor and the microenvironment. We will talk about immune cells and ability to understand different subtypes of cancer. We'll talk about macrophages, how the complex immune responses could actually be both detrimental and supportive of tumor development. And we'll also talk about early stage cancer detection, like biomarkers in exosomes. So a number of different types of projects that we will look at. We'll offer also a collection of curated data sets that you can kind of try to apply some of the methods that we went over in the program to something that is more relevant to maybe your research interests. And we did call this transcriptomics for chronic diseases. So those are the types of examples that we're going to use. But if you would like to then use it to a data set of your own, that is also going to be possible. And we have several ways that you can do that, including talking to someone that can guide you through that process, discuss your project with you, and help you kind of arrive at those results. So again, if any questions about this program, happy to answer them now. If you have questions that you would like to discuss after the session, just please email us hello@pine.bio. We'll schedule a time and we'll talk about your project as well. The next program I would like to talk about is called Bioinformatics for Infectious Diseases. And this is the application of biomedical data analysis to study outbreaks, for example, of epidemics, like we saw in this pandemic, but also how to connect it to genomic detail in the pathogen and an understanding of how the pathogen enters the host and what is the response of the immune system. So again, Raquel, to your question about being able to access this recording, after this webinar, you will receive an email with the recorded version, or you can go and look Pine Biotech, look us up on YouTube, and the recording is going to be there on our channel. So in the context of bioinformatics for infectious diseases, we will look at a few different specific methods, right? First of all, we will look at sequence variation and alignment, how we can use alignment to each other to understand differences of genomic sequences between themselves, or how do we organize them into groups using evolutionary and phylogenetic analysis. We will also look at discovery of variants and association between variants and specific changes, for example, at the protein level or at the population level. And then finally, we'll touch on transcriptomic analysis of host response, primarily around infected, uninfected, mock-infected types of experiments where we can understand the host responses at the cellular and tissue level. So the beginning of the program will start by kind of comparing and contrasting infectious diseases with non-infectious diseases. We will then talk about different pathogens, bacteria, viruses, parasites, and we'll talk about how they cause disease. What is it that we actually call infectious disease? How does it manifest? What are the specific things that we look at? And how can we analyze those with different types of molecular indicators? And so that will allow us to understand, using again some case studies, how a number of different types of could be used to characterize disease and understand disease progression as well as outcomes. And that will be kind of the main idea of this program, is it's multi-omics data in the context of infectious diseases. So the I think the idea here has become, infectious diseases have become one of those examples where these advanced new technologies like next generation sequencing have been used extensively to almost in real time track the development of an outbreak of an infectious disease and also characterize in very great detail how the pathogen interacts with the host in order to address that problem and create a diagnostic, create a panel or a simple process that could identify a very specific strain of a pathogen that we now know better how to deal with. Also, it has been used to identify the types of vaccines, for example, that we would like to design, or an intervention like an antiviral or a drug that will interfere with that process of infection. And so using these kinds of case studies, we're going to take a look at how does that translate to actually understanding the methodology. What do we do with data? What kinds of data? And how do we actually practically make use of that data to arrive at such conclusions? So again, I talked a little bit about the different types of methods that we will cover, and importantly, it will come with several different examples, project examples that you can try. SARS-CoV-2 genomics, of course, Ebola virus epidemic, as well as malaria infection. And again, this will be primarily around genomic and transcriptomic data. Farhana, I think Parisa here has a question on how to register in iLab. So if you could please share that with her. So finally, I want to talk about a program that is not specific to a type of data or a type of an example, but is more focused on the project that you would like to complete during this time. And this is applicable to two types of people. So number one, researchers, whether you are an undergraduate, high school student, or faculty. If your goal is to perform research and you would like to find a specific data set, analyze that data, and come out with a poster presentation or a publication, this is what this program is going to be designed around. And the second example of someone is maybe someone who would like to understand the process of research. So one, you know what kind of research you'd like to do. You have a goal and you know how to more or less get there. You need the help with bioinformatics. Or the second one, you don't know what research looks like, and you would like to understand how to perform research, but using bioinformatics because maybe you don't have access to a wet lab, or maybe you are just trying to maybe do two things at the same time. So the research fellowship is really around the process of kind of determining what your research question is, finding a data set that can help you address your research question, and then being able to address the kind of question that you have with the evidence identified in a particular data set. And the enablement that we offer here is big data analysis infrastructure. So we will support your big data analysis needs. If you need to analyze a data set with 10,000 samples and you can't do that on your computer, you will be able to do it here. mentorship. So someone will work with you to identify a question, refine that question, make sure that that question could be answered with the data that you've selected, and then help you get there. And along the way, we will provide you with opportunities to train yourself to be able to do this independently. So instead of this being training first, training is embedded into that process, because ultimately, if you don't know how to perform a particular type of analysis, you can just ask someone to do it for you. Or here, you will learn how to do it on your own and be able to apply it to that project. And we've selected a number of directions on how you can actually do this in the context of precision medicine, population health, biotechnology and agriculture, or space and astrobiology. So the first step here is going to start with an overview of the general background, how to do literature review, identify different things that people have already done to select something that is still unanswered, or maybe there's a gap in research, or maybe a debate and controversy, and how to use that process of literature review to be able to identify a very specific question that you would like to focus on. Once we do that, then we have a very well-defined process that will include weekly meetings, submitting a form about your progress, keeping a journal of your progress from learning to identifying a research project to then conducting the analysis, and eventually working with the community of both mentors and other participants to go through that whole process and submit the project. And the project will first of all be submitted on the portal. So on the portal, after you complete your profile, and you know that is free, you can just complete your profile and update it. You will then be able to start working on a project after you have 10,000 points. And so that's where the training comes in. So you still, in the context of this program, will have to go through some training. And importantly, we have another project that is designed to guide you through the different steps of literature review, identifying data sets on public repositories like NCBI, and then working through your analysis. Once you have done that, you will be able to fill in sections of a project from title to abstract to methodology, etc. And along the way, get guidance from our mentors that will help refine and kind of accelerate that process for you. And we have a number of individuals that have completed the research fellowship. They have either submitted a preprint in the course of the 3 months of that program. Some of them have went on to then publish. So again, I'm talking about undergraduate students here, or even have used their results in an ongoing research study that they're involved in, or just generally use this.
Right, guys, let's wait for Elia. I believe he got disconnected.
I mean, sorry, don't know what happened. But in any case, I was close to the end. So if anyone has any questions about these programs, going to be available to answer those questions or meet with you to discuss your specific objectives. We do have a couple of minutes left here, so if you do have any questions here, I'd be happy to answer those. And Parissa here is saying that you qualify through IDEA. So again, I want to briefly show you what will happen once you are registered. Okay, so let's say, for example, you are interested in biomedical data analysis using R and Python. Yes, great question, Raquel. How do you know? So this is a page where, again, I'll just go to my profile. So this is my profile, and you see that under badges, I have a number of programs that I have been added to. And I also have a starter subscription. So once you pre-register, right, the pre-registration was when you filled out the form and you said, you know, I'm from this university, this is my degree, this is which program I would like to join. Once you fill out that form, you are now ready for registration. And then JAPS or someone else will reach out to you, ask specific questions. If you do qualify, you will receive an email. So you'll have between now and June 15th to be fully registered. And how do you know that you have been registered? You've received an email. And for that, please make sure that you have our contacts in your contact book so that they're not going to spam. Those include hello@pine.bio, support@pine.bio. So they're all going to be pine.bio accounts. And they will notify you that you have been accepted. And then you will see a badge right here with the description of the program that you are in, and you will get a starter subscription. So if you go to your profile, again, how to get to your profile, you go in the menu and click on your email address. And then you will see a program page. And the program page, this is a program page that I am not a part of. So here you will see request more information or start your application. And if you scroll down, you will see here pricing. But if you have been accepted, you're not going to see pricing anymore. And I will show you that in a second. But this is an example of a program page. It has a schedule of sessions. In each session, you have additional resources that you can complete. And it's got a full overview of these right here on top. If I go to a page where I have, where I'm already a member of that program, I'll just go to this one. In here, I am already a member. So I will see it like this. And here you can see that I see my personal progress. I have access to the coursework. And instead of just seeing a schedule, I see the session video if it already passed, that's a previous one. Or for future ones, I will see a Zoom link. And I can click on that Zoom link and actually participate in that Zoom on the specific date and time. So here, I can also complete sessions. And once I complete them, once I complete all of them, I will get the certificate for completing that program. Any other questions? Okay, so if you qualify through LBRN, do you register for the classes via iLab? Farhana, I think that is for you. I think for LBRN, you actually, yes, for LBRN, you would have to go to this link. To this registration link. Again, this is just for people who are from LBRN. And once you submit this form, you will be evaluated by Dr. Ramesh Subramanian, who is the program coordinator, and he will, or director, and he, if you are approved, then you will receive an email notification from either Ramesh Subramanian, Farhana, or John Cubido. Okay, so Adeyemi, you just joined. We had a full webinar already. We're just about to, by when are you supposed to get the, oh, okay, sorry, Farhana. Farhana, would you like to go over this again?
Yeah, can you repeat the question?
There are two questions. So the first one is, if we qualify through LBRN, do we register for the classes via iLab? And the second one was, by when are we supposed to get the notification?
Okay, so if you are part of the LBRN, the form that you can see right now here on the screen is the red cap application form, I guess. Yeah. So just fill out this, and if you are approved, you will be notified by John, I guess. So they have some criteria. It's the registration is free for a limited number of LBRN members. So not all of LBRN members will be actually getting the free registration offer. So John and Ramesh will be looking at your application, and if you qualify, then you will be approved and they will notify you. And if you are approved, you will get an email from me explaining the iLab registration process. So far I already got 20 approved LBRN members, either faculty or staff or students. So I guess the registration process is over, but you can still contact and try to fill out this form, see whether they have any more spots available. So yes, first pre-registration through this registration form, and then fill out the forms in iLab to get your registration completed. So 2 steps. So that's all, right? Hello?
Thank you, Dr. Farhana.
You,
I guess,
can you hear me?
yes, I hear you.
Yeah. Okay, so the answer was clear, right?
Yep.
Okay.
Thank you for those information. And I guess that wraps up our presentation today. We would like to thank you for to each and everyone who has taken their time to join the webinar today. Feel free to reach out if you have any further questions. And for those who are interested, feel free to email hello@pine.bio. And also we will send a follow-up email here shortly with the link for for today's webinar. Thank you, guys.